The overall goal of this procedure is to introduce a way to study the functional integration of the adult-born dentate granule cells. This is accomplished firstly by stereotaxic injection of retrovirus into the dentate gyrus of adult mice to label the newborn dentate granule cells. The second step of the procedure is to perfuse the mice at certain time stages with cold cutting solution and remove the brain. The third step of the procedure is to prepare acute brain slices with a vibratome. The fourth step of the procedure is to perform whole cell patch clamp recordings on newborn dentate granule cells and stimulate the molecular layer of acute brain slices. Ultimately, results can be obtained that show functional synaptic integration of newborn neurons by recording and analyzing evoked postsynaptic currents on newborn dentate granule neurons.
The main advantages of this technique are retroviral labeling of newborn neurons in the adult brain and acute brain slice preparation from the adult animals. This method can help answer key questions in the field of adult neurogenesis, such as the functional roles of newborn neurons in the adult brain.
Prepare for the virus injection by removing the retrovirus for green fluorescent protein GFP or red fluorescent protein D tomato from the freezer and thawing it on ice. Thaw 2 microliters of retrovirus per animal. Anesthetize a 4 to 6 week old female mouse. Test for anesthesia by pinching the paw. Mount the mouse on a stereotaxic frame. Shave the hair on the head and wipe the skin with 70% ethanol. Use a scalpel to cut the skin and expose the skull. Adjust the nose position of the mouse so that bregma and lambda are in the same horizontal plane. Using a dental drill with a 0.6 mm drill bit, drill 4 holes through the skull at the coordinates shown here. Quickly fill a 1 microliter Hamilton flat-tip syringe with the defrosted retrovirus. Position it above one of the holes created at -2 mm bregma. To inject the retrovirus into the dentate gyrus, lower the syringe to a vertical depth of 2.5 mm. Inject 0.5 microliters of retrovirus at a rate of 0.25 microliters per minute. To prevent fluid backflow, wait 2 minutes after each injection before slowly withdrawing the syringe. Briefly rinse the external surface of the syringe tip with sterile PBS and an applicator to remove traces of blood. Use the same procedure to inject 0.5 microliters of retrovirus into the other 3 sites at ventral depths of either 2.5 or 3.2 mm. Close the wound with sterile surgical suture material and return the animal to its housing until the newborn neurons have reached the desired maturity date. After 21 days have elapsed, prepare for slicing the mouse brain by making the necessary solutions. Chill the cutting solution on ice at 0 to 4 degrees Celsius and bubble it with 95% oxygen, 5% carbon dioxide for at least 30 minutes. Place the artificial cerebrospinal fluid, or ACSF, at room temperature and bubble it with 95% oxygen, 5% carbon dioxide for at least 30 minutes. After 30 minutes on ice, set up the ice-cold oxygen-saturated cutting solution for cardiac perfusion. After anesthetizing the mouse, perform cardiac perfusion. Then, quickly remove the brain and place it in a petri dish containing cutting solution and a piece of filter paper. In the petri dish, cut off the cerebellum and then make a coronal cut 1 to 2 mm anterior to the injection sites to make a flat surface for mounting. Superglue the brain onto a vibratome platform. Mount the platform into the vibratome's cutting chamber filled with oxygenated cutting solution. Lower the blade and set the cutting window on the vibratome. Begin slicing the tissue. Cut each slice into 2 halves along the midline. Use a large bore pipette to transfer these slices to an incubating chamber containing room temperature ACSF, bubbled with 95% oxygen, 5% carbon dioxide. Continue slicing until the anterior end of the hippocampus is visible. Incubate the slices bubbling continuously in a 32 degrees Celsius water bath for 30 to 60 minutes. Return the incubating chamber to room temperature for the duration of the recording. The slices are now ready for recording. Begin by perfusing the recording chamber with 32 to 34 degrees Celsius oxygen-saturated ACSF at about 2 milliliters per minute. Use a large bore pipette to transfer a slice into the recording chamber. Place a U-shaped platinum wire with nylon strings on top of the slice to hold it down. Under low magnification, identify the molecular layer of the dentate gyrus. Then, lower the stimulation electrode into the molecular layer of the dentate gyrus. Under high magnification, identify the newborn dentate granule cells in the subgranular zone or granular layer by the expression of the green or red fluorescent protein labeling. Locate newborn neurons expressing GFP or D-tomato by shifting between DIC and fluorescent filters. Select a cell with relatively bright fluorescence and a healthy cell body for whole cell patch clamp recording. Insert a glass micropipette with a 3 to 6 megaohm tip resistance filled with the appropriate internal solution into a micropipette holder on the amplifier head stage. Under the high magnification objective of the microscope, lower the micropipette by a micromanipulator to the selected dentate granule cell and form a whole cell patch. Inject a series of currents under current clamp mode and record the firing properties of the newborn neuron. Apply single pulse or paired pulse electrical stimuli via the stimulation electrode and record the evoked postsynaptic currents. If there is no postsynaptic response, move the stimulation electrode to another position in the molecular layer and try to record the postsynaptic currents evoked from the new stimulus location. After finishing the experiment, use clamp fit to measure the amplitude of the evoked postsynaptic responses from the newborn neurons. This figure shows GFP expression in newborn neurons in the dentate gyrus of an adult mouse, 21 days post-injection of GFP retrovirus. Note that the newborn neurons are easily visualized and that both the dendrites and axons are robustly labeled. The dendrites are the processes that extend from the soma into the molecular layer of the dentate gyrus, and the axons are also visible in the hilus and CA3 area. This figure shows whole cell current clamp recordings from a newborn dentate granule cell at 21 days after injection of PUX-GFP retrovirus in an adult mouse. The top trace shows action potentials elicited by current injection. This figure shows postsynaptic currents elicited in a newborn dentate granule cell 21 days after virus injection by stimulating in the molecular layer. The existence of the postsynaptic response shows that the newborn neuron has already integrated into the existing neural circuits and is receiving synaptic inputs from afferents of various origins.
After its development, this technique paved the way for the researchers in the field of adult neurogenesis to explore the functional integration of newborn neurons in the adult brain.